Hello everyone. Welcome back. Today, we'll be discussing embryology of the urogenital system part 2. The urogenital system develops from the intermediate mesenchyme, mesoderm, the urogenital ridge. The part of the urogenital ridge giving rise to the urinary system is the nephrogenic cord, and the part giving rise to the genital system is the gonadal ridge. The genital system. Chromosomal sex of an embryo is determined at fertilization by the kind of sperm, X or Y, that fertilizes the oocyte. Male and female morphologic characteristics do not begin to develop until the 7th week. The gonads, testes and ovaries are derived from 3 sources, which includes the primordial germ cells, the intermediate mesoderm, genital ridge, and the mesonephric cells. The initial stages of gonadal development occur during the 5th week, when a thickened area of mesothelium develops on the medial side of the mesonephros. Proliferation of this epithelium and the underlying mesenchyme produces a bulge on the medial side of the mesonephros, the gonadal ridge. Finger-like epithelial cords, gonadal cords, soon grow into the underlying mesenchyme. The indifferent gonad now consists of an external cortex and an internal medulla. In embryos with an XX sex chromosome complex, the cortex of the indifferent gonad differentiates into an ovary, and the medulla regresses. In embryos with an XY sex chromosome complex, the medulla differentiates into a testis, and the cortex regresses. During the 6th week, the primordial germ cells enter the underlying mesenchyme and are incorporated in the gonadal cords. Development of testis. Testis determining factor induces the seminiferous cords to condense and extend into the medulla of the indifferent gonad, where they branch and anastomose to form the rete testis. Gradually, the enlarging testis separates from the degenerating mesonephros and is suspended by its own mesentery, the mesorchium. The seminiferous cords develop into the seminiferous tubules, tubuli recti, and rete testis. The seminiferous tubules are separated by mesenchyme that gives rise to the interstitial cells called the Leydig cells. By the 8th week, these cells begin to secrete androgenic hormones, testosterone and androstenedione. In addition to testosterone, the fetal testes produce a glycoprotein, antimullerian hormone, AMH, or Mullerian inhibiting substance, MIS. Antimullerian hormone suppresses the development of the paramesonephric ducts, which forms the uterus and uterine tubes. The walls of the seminiferous tubules are composed of 2 types of cells, the Sertoli cells and the primordial sperm cells, spermatogonia, derived from the primordial germ cells. Development of ovaries. Gonadal development occurs slowly in female embryos. The ovary is not identifiable histologically until approximately the 10th week. Cortical cords extend from the surface epithelium of the developing ovary into the underlying mesenchyme during the early fetal period. As the cortical cords increase in size, primordial germ cells are incorporated in them. At approximately 16 weeks, these cords begin to break up into isolated cell clusters known as the primordial follicles, each of which contains an oogonium, derived from a primordial germ cell. Development of the genital duct. During the 5th and 6th weeks, the genital system is in an indifferent state, and 2 pairs of genital ducts are present. The mesonephric ducts, Wolffian ducts, which play an important part in the development of the male reproductive system. The paramesonephric ducts, Mullerian ducts, which have a leading role in the development of the female reproductive system. Testosterone stimulates the mesonephric ducts to form male genital ducts, whereas Mullerian inhibiting substance causes the paramesonephric ducts to regress. Under the influence of testosterone, each mesonephric duct becomes the epididymis at the proximal end, and the efferent ductules in addition to a muscular ductus deferens at the distal end. The mesonephric ducts of female embryos regress because of the absence of testosterone. The paramesonephric ducts develop because of the absence of Mullerian inhibiting substance. Female sexual development during the fetal period does not depend on the presence of ovaries or hormones. Later, estrogens produced by the maternal ovaries and the placenta stimulate development of the uterine tube, uterus, and the superior part of the vagina. The paramesonephric ducts form most of the female genital tract. The uterine tubes develop from the unfused cranial parts of these paramesonephric ducts. The caudal fused portions form the uterovaginal primordium, which gives rise to the uterus and the superior part of the vagina. The endometrial stroma and myometrium are derived from spikemate mesenchyme. Auxiliary genital glands in females are outgrowths from the urethra into the surrounding mesenchyme, and they form the urethral glands and paraurethral glands. The fibromuscular wall of the vagina develops from the surrounding mesenchyme. Development of the vagina extend from the urogenital sinus to the caudal end of the uterovaginal primordium. The sinovaginal bulbs fuse to form a vaginal plate. Later, the central cells of this plate break down, forming the lumen of the vagina. The hymen usually ruptures during the perinatal period and remains as a thin fold of mucous membrane just within the vaginal orifice. Development of the external genitalia. Up to the 7th week, the external genitalia are similar in both sexes. Distinguishing sexual characteristics begin to appear during the 9th week, but the external genitalia are not fully differentiated until the 12th week. Early in the 4th week, proliferating mesenchyme produces a genital tubercle, primordium of penis or clitoris, in both sexes at the cranial end of the cloacal membrane. Labioscrotal swellings and urogenital folds soon develop on each side of the cloacal membrane. Masculinization of the indifferent external genitalia is induced by testosterone. During the 12th week, a circular ingrowth of ectoderm occurs at the periphery of the glans penis. When this ingrowth breaks down, it forms the prepuce, foreskin. The corpora cavernosa and corpus spongiosum of the penis develop from mesenchyme in the phallus, forms penis. The labioscrotal swellings grow toward each other and fuse to form the scrotum. The line of fusion of these folds is clearly visible as the scrotal raphe. The primordial phallus in the female fetus gradually becomes the clitoris. The clitoris is still relatively large at 18 weeks. The urethral folds do not fuse, except posteriorly, where they join to form the frenulum of the labia minora. The unfused parts of the urogenital folds form the labia minora. The labioscrotal folds fuse posteriorly to form the posterior labial commissure, and anteriorly to form the anterior labial commissure and mons pubis. Most parts of the labioscrotal folds remain unfused, but develop into two large folds of skin, the labia major a. Testicular descent. Testicular descent is associated with enlargement of the testes and atrophy of the mesonephroid, which allows movement of the testes caudally along the posterior abdominal wall. Atrophy of the paramesonephric ducts induced by the Mullerian inhibiting substance, MIS, enabling the testes to move transabdominally to the deep inguinal rings. Enlargement of the processus vaginalis, which guides the testis through the inguinal canal into the scrotum. Ovarian descent. The ovaries also descend from the lumbar region of the posterior abdominal wall and relocate to the lateral wall of the pelvis. The gubernaculum is attached to the uterus near the attachment of the uterine tube. The cranial part of the gubernaculum becomes the ovarian ligament, and the caudal part forms the round ligament of the uterus. Clinical correlates of the genital system includes mesonephric duct remnants in males, mesonephric duct remnants in females. Since the mesonephric ducts regress in females, remnants may remain as vestigial structures like the epiphyrin, periphyrin, and Gartner's duct, which can sometimes form cysts. Paramesonephric duct remnants in males. The The paramesonephric Mullerian ducts normally regress in males under the influence of anti-Mullerian hormone. Remnants may persist as the appendix testis or prostatic utricle. Androgen insensitivity syndrome, EIS. This is a genetic condition where cells are unable to respond to androgens due to receptor defects. Leads to varying degrees of undervirilization. Complete. EIS results in phenotypically female individuals with undescended testes and absent uterus. Mixed gonadal dysgenesis. This is a disorder of sex development characterized by asymmetric gonadal development, example, one testis and one streak gonad. Hyperspadias. This is a congenital anomaly where the urethral opening is located on the ventral underside surface of the penis rather than at the tip. Epispadias. In this anomaly, the urethral opening is located on the dorsal upper surface of the penis. It is often associated with bladder exstrophy, agenesis of external genitalia. In this condition, there is complete absence or failure of development of the external genital structures, the penis, scrotum, clitoris, and labia. Bifid penis. This is a congenital malformation where the penis is split into two halves, partially or completely. It is caused by incomplete fusion of the genital tubercle during embryogenesis. Micropenis is a normally formed penis that is significantly smaller than average for age and stage of development. Anomalies of uterus includes uterus didelphys, double uterus, absence of vagina and vagina atresia. In summary, the urogenital system develops from the intermediate mesenchyme, mesoderm. Urogenital ridge gives rise to the urinary system and the genital system. The urinary system gives rise to kidney, ureter, bladder and urethra, while the genital system gives rise to the gonads, ovary, testis, the genital ducts and genitals.
If you found this helpful, make sure to like, share, and subscribe, and I'll see you in the next video.